Hi, my name is Sarah Tucker, and I am a second-year PhD student at the University of Hawaii. And one thing that I found really great about the course was that after the first week of learning how to do enrichments for certain types of microbes and to be very selective in the medias that we use, so knowing what our electron donors are, our electron sievers are, and be able to select for certain microbes, we were given the task of, as a group, going out and enriching for a certain microbe. In my group, we were looking through this 16S amplicon data that we had from Trunk River, so it was allowing us to see which microbes were there. And we ended up clicking upon one group that was called Geobacter. Now Geobacter, they're a really, really cool microbe. They can produce electricity, and I don't normally think of them just being in a river system. I think of them for uses with fuel cells and other types of bioremediation. And so it was really cool for us as a group to be like, at that moment, we're going to try and get Geobacter. And the way that we made it really selective is the fact that Geobacter can have its electron donor outside of its cell. That's how it creates this electricity. And so what we did was we only had an electron donor that was outside of the cell in the manganese oxide form. And so this was just a super, super selective way of getting this organism. And as a group, each one of us came from a very different background. We had two chemists in our group, two environmental microbiologists, and one more of a bioinformatician. And so each one of us played a role in that experience. And at the end, we were just so happy to be moving forward towards this really cool microbe that has so many great implications for technology, but also can be found right down the street in Trunk River.